That's the list maybe.
So he's on video, she's there,
He's,
That's him,
that
right?
was him.
He's on video.
Video and
This one.
perfect.
Or maybe
Take off.
is it okay with you?
Yeah, absolutely.
Hi everyone. Hi, thanks for being here in Glasgow and remotely from home. Um, we're going to have a great session this morning from the Womb to Wisdom. My name is Anne Gallagher. I'm from University of Montreal, Canada, and I'm co-chairing this session with my colleague Jürgen Lukart from the Research Institute Jülich in Germany. So we're going to get 6 talks, about 10 minutes each, and a few minutes of questions. And please, for the questions, use the Q&A tool on the OHBM application or on the website. So let's start right away with the first talk. We have a couple of talks on video, 2 of them, and the other speakers are here. So for the first one is on video. That's a talk from Kyo Im, and it's on the association between placental oxygen transport and cerebral cortical growth in the fetal brain. Thank you.
In this presentation, I will talk about association between placental oxygen transport and cerebral cortical growth in fetal brain. Human fetal brain shows very dynamic development process and major neurogenic event, including neurogenesis, proliferation, and migration. We can see very rapid brain cortical surface growth and folding with cortical circuit and axonal development. Because normal cortical growth and folding patterns are important for normal brain function, it's been of great interest to understand developmental mechanism of human cortical growth. Human cortical development is strongly controlled by genetic process. Cortical functional areas and folding patterns are specially associated with gene expression pattern in ventricular zone and cortical plate. But gene expression associated with brain development can be modified by environmental factors in fetal life. Our previous study and data also showed that genetic is not everything in brain development. When we see 3D brain surface model in monozygotic twins who have the same genotype, their brains look different. They are more similar than unrelated pairs, but their brain structures are still not identical. Even before birth, we can see different brain shape in fetal monozygotic twins, so there must be many other factors influencing fetal brain cortical growth. Among many factors, placental function is a crucial factor for fetal growth. Fetal health is dependent on placental function, especially placental transport of oxygen from mother to fetus. Placental insufficient oxygen transport can result in fetal growth restriction and atypical cardiovascular, metabolic, and neurological development. And this atypical fetal growth can also affect brain development. So our hypothesis is that placental transport of oxygen may be one of the important non-genetic factors that affect early human brain growth. And this study aimed to assess placental oxygen transport function and examine its impact on fetal brain cortical development in vivo using fetal MRI. We performed monochorionic twin brain analysis because twin subjects identical genotype and gestational age. So we examined the placental transport effects controlling for genetic and maternal factors. To present our study, first I will show in vivo MR imaging of fetal brain structure and placental transport function, and I will talk about our quantification of fetal cortical growth and folding pattern and placental oxygen transport. Then I will show significant relationships between cortical growth and placental function. Our study included 8 monochorionic twin pairs, and their age ranged from 27 to 34 gestational weeks. We scanned fetal brain using single-shot fast spin-echo T2-weighted imaging for structural MRI. It is usually necessary to acquire multiple images in axial, coronal, and sagittal views due to interslice motion artifact. To image placental oxygen transport function, we are using bold MRI. We perform bold MRI with maternal hyperoxia, which is transient 100% oxygen inhalation, and it can non-invasively measure blood oxygenation in the placenta. We can see imaging signal change during hyperoxia in placenta region. To quantify fetal cortical growth, we process structural fetal MRI data using our pipeline. From the raw data, we use the UNet deep convolutional neural network to automatically segment only brain regions, and we excluded bad quality imaging data for further reliable data process. And T2-weighted structural fetal MRI shows interslice motion artifacts, so we are using slice to volume matching and registration techniques to remove that interslice motions. After motion correction, we performed cortical plate segmentation. We recently developed automatic fetal cortical segmentation method using 2D fully connected convolutional neural network. From the segmentation volume, we reconstructed the surface model with the same number of triangles and a good smooth mesh property. For whole brain global analysis, we measured cortical volume, cortical surface area, gyrification index, and mean sulcal folding depth in the left and right hemispheres. We performed sulcal regional analysis. Recently, we developed the method for automatic postulation and labeling of cortical sulci in the fetal brain. Using our automatic sulcal labeling method, we measured surface area and sulcal folding depth for each sulcus. We also analyzed the global sulcal folding pattern, which is the patterns of arrangement, number, size, and relationship of sulcal fold. It's biologically meaningful to examine the sulcal folding pattern because it is related to size, organization, and spatial arrangement of cortical functional areas and their white matter connections. And this sulcal pattern variability is related to several human functions and many neurological disorders. For quantitative sulcal pattern analysis, we characterize the sulcal pattern with the graph structure, and this graph has geometric and topological features of sulcal fold. Previously, we developed a method to compare sulcal graphs between different brains using a spectral-based graph matching technique. Then we calculated the normalized sulcal pattern similarity index using combined features of position, depth, and area of sulcal fold. And this index ranges from 0 to 1. We can also use a single feature and measure the sulcal position, depth, and area patterns separately. For quantification of placental oxygen transport, first we manually delineated the placenta regions corresponding to each twin fetus and extracted placental signal change with methanol hyperoxygenation. We employed the gamma function to capture and model the signal change and obtain time to plateau TTP value after fitting the signal to the model. TTP can be a surrogate marker assessing regional oxygen delivery function in the placenta. For healthy subjects, TTP values were short and placental histology was normal, but for pathological cases, TTP values were longer and less uniform and histology was abnormal. We statistically analyzed the relationship between mean placental TTP value and global and sulcal regional cortical growth measures and sulcal pattern similarity between twin pairs. Volume, surface area, gyrification index, and sulcal depth were compared between twins with low TTP and twins with high TTP using a paired t-test. For sulcal pattern analysis, we examined the relationship between sulcal pattern similarity and TTP difference of twin pairs using correlation analysis. The group analysis showed that fetuses with long TTP value show significantly lower cortical plate volume in the left and right hemispheres. And cortical surface area in the left and right hemispheres were also significantly reduced in the fetuses with high TTP values. Whole brain mean sulcal depth and gyrification index were not statistically different. Our sulcal regional analysis found significantly reduced sulcal depth in the right inferior frontal sulcus and right parieto-occipital sulcus in fetuses with high TTP value. In the sulcal pattern analysis, sulcal pattern differences were significantly correlated with TTP differences in twins in the right hemisphere. When TTP difference was larger, sulcal pattern similarity was lower between twin pairs. And we examined each feature separately and found sulcal position and depth patterns were associated with TTP value. In conclusion, we identified placental non-genetic prenatal influences on human cortical development. Alterations in the placental oxygen delivery function caused reduced volumetric and surface growth of the fetal cerebral cortex and altered sulcal folding pattern. And these cortical structural alterations may be associated with the future neurodevelopmental outcomes at the birth. So this study provided meaningful information for understanding developmental mechanisms of the human cerebral cortex. Thank you for your attention.
Okay, because the speaker is on video, we will not be able to ask questions for this talk. So we move on to the second talk by Valérie Sidnon from University of Pennsylvania, and she will talk about developmental refinement of spontaneous activity, varies across sensory, motor, and association cortices.
Great, thank you. Hello, OHBM. My name is Valérie Sidnor. I am a neuroscience PhD student at the University of Pennsylvania in Ted Satterthwaite's lab, and I'm really excited to be here today to tell you about how the developmental refinement of spontaneous activity varies across a sensory motor to association cortical axis, and how this provides new insight into the spatial and temporal sequence of human neurodevelopment. So human cortical development is a dynamic, protracted, and heterochronous process. During childhood and adolescence, different cortical regions maximally develop and mature at different ages. Elucidating the temporal sequence of human neurodevelopment, or in other words, the spatial progression of developmental plasticity across the cortical landscape, has profound implications for understanding mechanisms underlying this plasticity in health and disease, age-dependent brain-environment interactions, and the optimal timing for youth interventions. Nonetheless, the precise spatiotemporal sequence of human cortical development has yet to be really systematically characterized. Now, extensive prior work has shown that major developmental changes happen earlier in sensory and motor cortices, shown on this brain in yellow, and later in association cortices, shown here in purple. This has been demonstrated with human neuroimaging for large-scale features such as cortical thickness, myelination, and connectivity, and by animal model studies characterizing the maturation of plasticity-regulating features such as parvalbumin interneurons. But critically, beyond this coarse division, there is additional temporal variability, yet a parsimonious framework that accounts for this variability is still being established. In a recent review article, I described one unifying framework which posits that during childhood and adolescence, cortical maturation progresses along a sensory-motor association axis. The sensory-motor association axis, or the SA axis, is an axis of brain organization that is globally arranged from primary cortices at the sensory-motor pole of the axis to modality-specific and multimodal cortices in the middle, and to transmodal cortices at the association pole of the axis. And this axis captures continuous and concerted variation in remarkably diverse features across the cortical sheet, including but not limited to features like cortical connectivity, cerebral blood flow, gene expression, laminar architecture, task-based activation, and evolutionary expansion. So in order to test whether this axis captures not only the spatial layout of cortical properties, but also the patterning of developmental change, we studied age-dependent changes in spontaneous cortical activity. Spontaneous or intrinsically generated activity plays vital roles in the developing brain, influencing, for example, neural migration, circuit wiring, sensory maps, synapse refinement, and myelination. Moreover, studies of the developing murine sensory cortex have repeatedly shown that as the cortex transitions from plastic to mature, spontaneous activity evolves from strong and more globally synchronized, as shown on the left, to sparser and decorrelated, as illustrated on the right. So we aim to non-invasively study this sort of stereotyped refinement in spontaneous activity in humans using resting state fMRI. And as we all know, fMRI is not a direct measure of neural activity. However, many studies employing simultaneous fMRI and electrophysiology or calcium recordings have revealed that a greater level of spontaneous activity and more synchronized activity will increase the amplitude of low-frequency bold fluctuations, suggesting that bold fluctuation amplitude may be higher when cortical plasticity is enhanced. We therefore predicted that as plasticity is reduced and the cortex matures during childhood and adolescence, that bold fluctuation amplitude would exhibit heterochronous declines along the cortex's SA axis. We tested this prediction in a cross-sectional sample of 1,033 youth, ages 8 to 23 years old, in the Philadelphia neurodevelopmental cohort. We quantified cortical bold fluctuation amplitude by calculating the average power in the time-varying bold signal in a 0.01 to 0.08 hertz frequency range. We then flexibly modeled associations between fluctuation amplitude and age within individual cortical regions using generalized additive models, which we used to fit an age spline while controlling for sex and in-scan motion. The first thing that we observed was that the magnitude and direction of fluctuation amplitude age effects varied across the cortex. We further visualize this variability by plotting fluctuation amplitude age fits in individual cortical regions, so each line in that plot is a region, which revealed this sort of cortical continuum of maturational trajectories. And in general, sensory regions like the primary visual cortex shown here tended to exhibit larger and earlier declines in fluctuation amplitude, which are potentially indicative of a decline in or decorrelation of spontaneous cortical activity. Transmodal association cortices, like the DLPFC, on the other hand, tended to show these inverted U-shaped curves, which are potentially indicative of heightened and synchronized activity in early adolescence. We next tested whether developmental change in bold fluctuation amplitude was related to the maturation of intracortical myelination, which is a very well-established plasticity limiting factor. To do this, we compared our data to results from Graham Baum and Leah Somerville's recent paper characterizing the maturation of the T1-T2 ratio in youth 8 to 21 years old. Shown here is that fluctuation amplitude and T1-T2 ratio overall age fits were very strongly inversely correlated, indicating that cortical regions that exhibit a larger increases in myelin content from childhood to adulthood also exhibited larger decreases in bold fluctuation amplitude. In addition, the age at which fluctuation amplitude began significantly decreasing in a region was associated with the age at which the T1-T2 ratio maximally increased. So this reveals temporal correspondence between fluctuation amplitude development and development of a main limiter of cortical plasticity. Having shown that fluctuation amplitude development tightly paralleled maturation of a plasticity regulator and broadly diverged between sensory motor and association cortices, we sought to determine if developmental patterns spatially conformed to the SA axis. To do this, I ran a PCA on regional fluctuation amplitude maturational trajectories. The first principal component is shown here, and this explained 87% of the variance in development of this functional measure and can therefore be conceptualized as the principal axis of spontaneous activity development. This principal developmental axis showed pretty striking homology to the SA axis, with principal developmental axis loadings and SA axis ranks correlating at R equals 0.7. Furthermore, if we take the SA axis and divide it into 10 bins and average regional age fits within a bin, you can see how the sort of spectrum of developmental trajectories visible at the regional level is recapitulated by SA axis ranks. Finally, we wanted to elucidate whether this sensory motor to association neurodevelopmental pattern was pronounced at every age between 8 and 23, or if it was most dominant in a specific age range. To do this, we quantified age-specific developmental effects at approximately 1-month intervals between 8 and 23 years of age. So here, for example, are age effects at 10, 15, and 20 years of age. We then correlated age-specific effects with SA axis rank. And this approach generates age-specific correlation values that quantify the extent to which developmental change is spatially ordered along the SA axis at a given developmental time point. Plotting age on the x-axis here and age-specific correlation values on the y, you can see that there's a robust alignment between developmental change and SA axis rank from around 8 to 17 years of age, with peak alignment between neurodevelopment and the SA axis observed at 15 years of age. Interestingly, this correlation rapidly dropped to 0 after age 18, suggesting there may be a program switch in the spatial patterning of subsequent developmental change after adolescence. So altogether, this work provides evidence that neurodevelopmental processes unfold along a sensory motor association axis from ages 8 to 18, suggesting that molecular timing mechanisms may pattern developmental change along this axis throughout the second decade of life, yet not after. Furthermore, our findings show that a main developmental axis is also a prominent spatial feature axis, suggesting that temporal developmental variability may produce spatial feature variability and providing developmental insights into adult brain organization. And then last, our data showed that during late childhood and early adolescence, transmodal association cortices seem to exhibit increases in the strength or coherence of spontaneous activity, which is a functional hallmark of plastic cortices. This aligns with theories that early adolescence may be a sensitive period of brain development characterized by heightened malleability in the association cortex, and thus indicates that educational, social, or psychiatric interventions may have a large impact on higher order areas of the brain in late development. And with that, I'd like to thank many people, including the entire Penn Lifespan Informatics and Neuroimaging Center, especially our really unstoppable informatics team, our collaborators on this project outside of PennLink, and then a special shout out to Bart Larson, who's a postdoc in Ted's lab, who basically functions as my scientific advisor and mentor and role model and statistical teacher. And then finally, thanks to Ted Satterthwaite, who doubles as my PI and my biggest cheerleader and supporter, and who is the absolutely best human to do science with.
Thank you very much for this fascinating talk. So far, I see only one question from the audience. The question is about myelination. Did you consider other myelination measures like MTR, magnetization transfer ratio, rather than T1 by T2?
Yeah, excellent question. I would love to look at MTR development data. We don't have access to that currently in the lab. So if anyone has MTR data and has ideas and wants to collaborate, please come talk to me.
May I ask you a question over here?
Yeah.
Do we have time?
Yes.
Okay, first of all, congratulations. That was a fabulous talk.
Thank you.
So the T1, T2 ratio, which is essentially a measure of, a surrogate for cortical myelination, you showed the high correlation. So what I wanted to know is, do you think that this spontaneous activation is independent of that, or is all you need T1 and T2? Because you can get 0.7 millimeter resolution now with that and do a very high definition cortical map of T1, T2 ratios. And as a corollary that, you showed this U-shaped thing happening in adolescence. Do you see that U-shaped curve in those T1 and T2 myelination, or T1, T2 ratio maps?
Okay, excellent questions. Let's see if I can address them all. So first question is, do we just need the T1, T2 ratio? Is that capturing all the information that spontaneous fluctuations are capturing? That's a good thought. I think there's definitely evidence from animal models that changes in myelination will dampen spontaneous activity, but I wouldn't think that's the only thing driving spontaneous changes, activity changes during development. There's also large changes in inhibition and excitation, and I would think that changes in EI balance above and beyond changes in myelination would be affecting spontaneous activity. So I think, yeah, future work looking at relationship between other things we know change during development and spontaneous BOLD would be of interest. I've forgotten the second question.
Do you see a U-shaped response to the T1, T2 ratios like you did in the spontaneous activation?
Okay, thank you. Great question. The answer is no. So T1, T2 ratio, or proxy for myelin, really just increases throughout development. Instead of doing inverted U-shaped curve, it might inverted U like across the entire lifespan. But what we do see in transmodal association cortices is that during early childhood, myelination is like flatter, it's not changing that much. And then once we get on the other half of the inverted U-shaped curve, when spontaneous activity or fluctuation amplitude is declining, that's when myelin starts to actually increase in transmodal association cortices.
Thanks a lot for this great talk. We have to move along. So we'll move along with the third speaker, Yankee Chou-Cartier-Dimer from Neurospin. She will talk about fold opening dynamics during the aging process, like new sulcus communities. Just give her a few seconds to plug your laptop.
Hello, everyone. I'm a PhD student from Neurospin, and I'm delighted to talk to you today about my work on fold opening dynamics during the aging process. My plan is fairly straightforward with a little contextualization first, then a quick overview of the course I'll be using. The goal here is to find out whether we can use a new brain feature to analyze a large-scale general population and compare it to a smaller cohort with only at-risk subjects. So aging is associated with morphological changes in the brain, which have been studied by many investigators using features such as cortical thickness, surface area, or gyrification index. It has been shown, for example, that with age, there is a reduction of gray and white matter and an increase in volume of cerebrospinal fluid. However, these changes are not uniform in the cerebral hemisphere. Moreover, numerous neurodegenerative diseases can cause accelerated brain atrophy. One of the main challenges of the field is to disentangle normal brain atrophy and due to healthy aging from the accelerated brain atrophy due to a disease. So the idea here is to rekindle the discussion by considering a new fault-related atrophy feature. To extract brain features, we used the brain visa. T1 weighted MRIs were processed to extract 60 sulci for both left and right hemisphere. And for each of the 120 total sulci, we can compute numerical measures such as the mean cortical thickness or the mean sulcal width. Notice that the regions of interest are not the same as in FreeSurfer. So why should we consider the sulcal width rather than the cortical thickness? First of all, one of the limitations of evaluating brain changes based on the gray and white matter segmentation is that the blurring between white and gray matter junction increases with age. And it is much easier to consider to measure the sulcal opening, which relies on the easier segmentation between gray matter in gray and the cerebrospinal fluid, which is in black on T1 weighted MRIs. This sulcal feature also yields better signal-to-noise ratio as it incorporates the atrophy of two cortical ribbons. On top of that, it is possible that the sulcal opening may unveil some other aging process, which is only present in the subcortical structure and not in the cortical thickness. For example, here we have the FLAIR MRI of an 80-year-old patient with extensive white matter hyperintensities, which are present here, but also here, which may affect the sulcal width of the neighboring sulci, but not the cortical thickness. Now let's take a look at the cohorts I used. For this study, we used two cohorts which are rather different. The UK Biobank database contains neuroimaging measures from 20,000 something subjects. On the other hand, the French Memento cohort study only recruited participants with isolated subjective cognitive complaints or presenting mild cognitive impairment while not demented. Besides, during 4 years of follow-up, 208 patients were diagnosed with dementia. So we call this the at-risk subject. One of the challenges will be to bridge the gap between the small and the larger scale population. Is there a link between UK Biobank and Memento? And if there is, why? To assess this link, we analyzed two image-derived phenotypes that we extracted from T1 weighted MRIs with brain visa. The cortical thickness, the mean cortical thickness, and the mean sulcal width for each sulci. First and foremost, we'll be working on the modelization of the age dependency on sulcal features of UK Biobank, which we consider here as a general population cohort. Let's dwell a little on the time trajectory of the cortical gray matter thickness of the right superior temporal sulcus. For each age group, I computed the median, in orange here, and the 90th and the 10th percentile, which means that at this group age, for the subject of this group age, 80% of these subjects have their sulcal width in this interval. I also added the age distribution of the population in overlay, which should help envision which values are more representative than others. So for the women here, it works. For the women here, you can see a kind of plateau here, and then an acceleration in the cortical thinning with the age, around this age. However, this trend cannot be observed for the men. The atrophy rate is rather regular, and is sensibly the same as the accelerated atrophy rate for the women. This may indicate the fact that men are subject to accelerated brain atrophy much earlier than women, probably due to more health risk factors. Now, looking at the sulcal opening for this same sulcus, this trend is a little more obvious. Again, only for the women. The sulcal opening is quite slow at the beginning, then it accelerates. And this acceleration in atrophy can be observed in many sulci, especially the big ones or the ones around the hippocampus area from which the Alzheimer pattern originates. While the trajectory of the cortical thickness of most of these sulci become highly noisy for elder subjects. Those, thus, will stick with the sulcal opening for the following. To identify points of acceleration of brain atrophy, a piecewise linear model with accelerated slope after a sharp inflection point provided the best fit on the sulcal opening of the women. The main difficulty here was to account for the high subject intervariability, because this is not a longitudinal study. That is why the model was fitted on a percentile line rather on all the data points. Moreover, the goal is to investigate whether we can whether there is a link between UK Biobank and Memento. That is why the model was fitted on the 90th percentile line, because we would want to focus on the at-risk subjects of UK Biobank. And these subjects should be the ones who have a wider sulci. Eventually, we chose the 90th percentile line because it should be low enough to eliminate most of the outliers and high enough to target the at-risk subjects. This model has 4 parameters: the slope and intercept for the first segment, and the slope and intercept for the second segment. And the tipping point age, the age of acceleration, is defined afterwards as the intersection of these 2 segments. For some sulci, this modelization just doesn't make sense. For example, the right orbital sulcus doesn't show any atrophy acceleration, even on the women. So, for each sulcus, a piecewise linear model was fitted, and the final values for the 4 parameters were set after computing 100 bootstraps to reduce overfit bias and to compute 90% in confidence intervals. As I said before, some sulci are not meant for this model. So how does this translate during modelization? Well, there are some sulci which present a decelerated brain atrophy rate after the breakpoints, which is probably due to other processes, but for now we'll put them aside because of our null hypothesis. Our piecewise linear model was supposed to capture an acceleration of brain atrophy only. So we only select sulci with positive slope change. Furthermore, we'll only select sulci which remained stable during bootstrapping. What does it mean? Well, this stability is quantified by the variability of the tipping point age during the bootstrapping. Indeed, when the training set changes ever so slightly, the 2 slopes change too. But if the 2 slopes were close one to another from the start, even a tiny variation would cause the tipping point age to be completely different. So, in other words, when the derived value, that is the tipping point age, remains stable during bootstrapping, that means that the point of acceleration is easy to find, so that the piecewise linear modeling makes sense. So here we have all the selected sulci, ranked by the size of confidence intervals during bootstrapping. Notice that the sulci with the most stable, with the most pronounced atrophy acceleration are almost all in the medial posterior temporal lobe and the occipital lobe. In the same spirit and studies focusing on structural cortical networks, we have performed spectral clustering of the parameters of the piecewise linear model in order to detect sulcus communities and, sulcus communities following the same opening trajectory during aging. Thanks to silhouette score, we found out that the optimal number of clusters is 9, and each color is matched to a cluster sulci that did not satisfy the null hypothesis or with too many missing values after pre-processing are in black. Now let's see if we can transpose the results we got from the UK Biobank dataset on the at-risk population dataset Memento. We chose to focus only on one of the memory tests, the free and cute selective reminding test, which we can call the long-term memory test. Here we computed a Pearson correlation between the so-called opening for each sulci and the memory test. The sulci are ranked, as before, by most stable modularization. In other words, the sulci which effectively show a neat acceleration in brain atrophy are roughly those which yield higher correlation with the long-term memory test. Now let's examine the clustering we did. Let's focus on the yellow cluster. There are both posterior lateral features, the right superior temporal sulcus, the left insula, etc. This yellow cluster yields better correlation with the long-term memory test than all the other clusters. And then for each cluster, we also took the pertaining sulci as features, and we fed them to an AdaBoost classifier to predict if a subject will progress to dementia in the next 4 years. With yet another process, the same yellow cluster shows better predictive power than all the other clusters. So what does this all mean? First, that sulcal opening is a better feature than cortical thickness to visualize and model brain atrophy. Second, that sulci with the most pronounced acceleration in atrophy rate are most correlated with long-term memory tests, and that can be captured by a model as simple as a piecewise linear model. Third, that even extracted from a general population, such as UK Biobank, some results can be transposed to an at-risk older population study, hinting at result reproducibility and complex physiopathological processes. So what next? Well, there are quite a lot of things left to do. For example, perhaps we can improve the modeling with the quantile normalization, or we could fit the piecewise linear model for each dementia subtype and identify aging trajectories to help patient prognosis. Well, I would be delighted to talk more about it at the poster session later. Thank you for your attention.
Thank you very much. So we have one question here in the Q&A. Could you predict brain age using the so-called width data?
Well, I tried during my internship, and no, it's too noisy. It doesn't work. I'm sorry.
Thank you very much.
Quick question, if it's possible. So I believe your result, when we read these scans clinically, and the AI tells us whether they have atrophy or not, they're only looking at cortical thickness, and you can see that they have atrophy, and it doesn't come up as cortical thickness, because you can see the sulci getting wider. So I think that's fascinating. But what do you think would happen? Like you mentioned memory loss, but frontal temporal dementia has a very different pattern of atrophy than Alzheimer's disease and other forms of vascular dementia. So how do you think your analysis would play out with that?
With other dementia subtypes, is that it?
Yes.
Yeah. Well, first, for now, I stuck with the Alzheimer's disease because it's the most common brain disease with a lot of data. I think it would be a little more difficult to work with other etiology because of the lack of large cohort.
Okay. Thank you very much again for this very nice talk. Also, just a comment to those who have questions, because we've seen that questions are posted to the previous speakers. Please approach the speakers directly, because we have 3 talks to go, and if you want to have additional questions asked, just approach them. So we have the pleasure to introduce the next talk by Mitim Yakov from the Karolinska University, who is going to speak about sex-related differences in multi-layer connectivity throughout aging. So.
Thank you for your introduction, and let me take the opportunity to thank the organizers of the conference for giving this opportunity to be here to share with you the research that we have of how sex influences the trajectories in functional connectivity throughout aging, and we did it via a new method of multilayer networks. So over the last decades, there has been an increase in the human lifespan, but unfortunately that has not been matched by corresponding increase in the health span. And in fact, the aging remains a major risk factor for many disorders. So it has been known now and it has been shown that not all individuals age at the same rate. And several studies have shown that in particular, sex seems to be responsible for some of this variability. And it's important to understand how and how sex influences these differences in aging, because this could potentially explain why there is different prevalence in several diseases between men and women. But also these studies in healthy aging, they could provide the benchmark across which all of the other studies that study some neurodegenerative disease could, for example, be compared with. Or if we know how sex and age really affect the functional connectivity, this could potentially fundamentally change how we treat them, in our statistical analysis, for example, as covariates. Unfortunately, the effect of sex has been understudied, and one of the potential reasons is that there is the availability of small samples only, which kind of diminishes the power, the statistical power of the study. So that's why in this study we have set out to do investigate this issue in a large cohort of around 35,000 subjects. And we did this with multi-layer networks construction, which will be the focus on the rest of my presentation. So to tell you several words about the data that we had. So we obtained the resting state functional MRI data from UK Biobank. They were pre-processed by the UK Biobank researchers. And the data that we got is the imaging phenotypes, which is 21 by 21 connectivity matrix for each participant. And you can see it here. So the nodes of this graph are basically the resting state networks. And the connections between them are the correlations between these networks. The positive correlations you can see here in red, while the negative or anti-correlations are shown in blue. Our data, we analyzed it for around 20,000 women and 17,500 men, which were at the age range of between 47 and 79. So moving to the second part of my talk and how we actually constructed this multi-layer network. So to start with, this is the data that we got. This is the weighted connectome of each patient with the positive and negative connections. And what we first did is that we split the positive and negative connections into two different networks. And this is the conventional analysis, what has been commonly done, where the negative connections are generally set up to 0, and only the positive connection connectome is analyzed. However, we thought that the interaction between these two networks could be important in order to understand these differences. So at the first step, we tried to integrate this in a multiplex network, which is basically a network of two layers. Each of these positive and negative networks becomes a layer in this one, and the interaction is defined by connections connecting the corresponding nodes. Then we can analyze this type of networks, and we can see how, for example, the patterns in the two layers are similar, how they overlap. And to do that, we chose, in particular, participation coefficient and clustering coefficient. Well, this is an analysis that has been done, and it has its advantages. It has one big disadvantage in a sense that the interaction between the two layers is fixed and it's unitary. So in order to address these limitations, we propose here a novel method which extends the multiplex to a multi-layer network, where we now don't have any restrictions on the connectivity between the layers, and every node can be connected with each one. And we also assign a weight to each of these interlayer connections so that we can analyze our result as a function of the weights. In our analysis, we actually saw that the weights, the results are pretty stable across the weight range of the interlayer connections that we considered. So I'm going to show only the results for only one. And of course, because this is a novel method, there are no measures present that will allow us to analyze the topology of these networks. So we actually designed two, the multi-layer global efficiency to assess the functional integration, and the clustering coefficient to assess the functional segregation of this network. And to explain how these measures are calculated, please consider this toy model, which consists of 2 layers, 5 nodes, and just the connection of one layer of one node is shown just for clarity. So at the first step, what one can do is one can discard the second layer and basically just calculate the global efficiency of the layer that the node is part of. And this is the standard calculation that has been done. But then in the second step, then what one can do is that one can create, we created a different network where we assess the interaction of this node with the nodes in the other layer, and we calculated the global efficiency of this network. So our measure at the end is the difference between the two, and this is a number that quantifies the global efficiency of this node. And then if we take the average of each node in the network, we get a single number that quantifies the level of functional integration in this type of multi-layer networks. And then, very analogously, similarly, by following the same procedure, we calculate the multi-layer clustering coefficient. So finally, very shortly, just to present to you several results that we obtained. So what you see here are the linear models that were fitted. Men are in blue and women are in red. And the models are shown also on the figure. And basically, the stars identify the ages at which men and women showed differences in that topological measure. So there are 2 things that are particularly important about this figure. The first one is that all of these multi-layer measures are able to capture differences in the topology between men and women. But also another important fact is that while the clustering coefficient tells us that these differences in functional segregation will be constant throughout the complete aging span, the multi-layer global efficiency shows an interaction between sex and age, and actually it shows that the differences in global efficiency are going to diminish as people get older, so that sex is not as important in the functional connectivity differences for older adults. And the second point is also more interesting when you consider the single layer and the conventional analysis that don't consider this interaction. You see that the clustering coefficient doesn't even find differences between men and women, and also these age-sex interactions cannot be captured by the conventional method. So this indeed tells us that the interaction between these 2 layers is something important, and it captures an effect that can show us some new insights. And then to just evaluate the relevance of these measures, we have run a partially square analysis with cognition, cardiovascular risk factors, and structural measures that in the UK Biobank sample that we had were significantly different between men and women. And then what we found out is that these factors, the multiplex and multi-layer measures, can explain a significant portion of the variance in these measures, furthermore showing the relevance of this integration between positive and negative connections. And just as a final word, we also ran a GWAS analysis in this sample, and we were able to, we found that these measures were associated with several genes that are implicated in processes that are shown to be changed with aging. So again, this furthermore solidifies this idea that the multi-layer measure indeed capture some aging-related effects. So I hope that in this presentation that I have convinced you that to study the interaction between the positive and negative connections is important, and indeed can really tell us something novel about how sex influences the functional connectivity throughout aging, something that cannot be captured by the analysis that are conventionally done until now. And finally, I would like to thank to everyone who has been involved in this paper, and especially the people who developed the BRAF2 software. All of the analyses that I showed you here were done with the software. So yeah, if you want to discuss more about it, or if you want to discuss about this work, if you have any questions, please visit at each of the posters. And with this, I would like to thank you for your attention. And if you have any questions, I would be happy to take.
Thank you very much for this very nice talk. So we have actually two questions, but time for one, so I will try to kind of combine the two. Why did you decide for a 21 network approach? Because isn't it too coarse? And how did you get the layer weights for comparisons?
The reason, so first of all, I didn't show it here, but the results are replicable also in the high parcellation networks that were provided by the UK Biobank. But the basic idea of using the 21 was that it was corresponding to a more well-defined resting state network. So, you know, the results, we felt that the results are going to be a bit more interpretable in that case. However, they are replicable in both parcellations.
Thank you very much. Oops, thank you very much. So in the interest of time, we'll go to our next speaker. We can give him a clap, sorry. We always have time for that. So our next speaker will be on video. So this is Addis Kalantar Ormozi from the Douglas Mental Health University Institute, McGill University. She's going to talk about multivariate analysis of cortical morphometry across human brain development.
Hello everyone, and thank you for the invitation. I'll be presenting my work on human brain development, a cross-sectional and longitudinal study integrating multiple neuromorphological features. So, studying the stages of brain development, it's been known for a long time that brain undergoes a massive cortical structural remodeling that starts from childhood, which continues well into the third decade of life. But surprisingly, looking at the huge body of the neurodevelopmental literature, most of our understanding of brain development is derived from studies examining brain development in the context of single morphological measures or studying them in statistical isolation, while ignoring the intrinsic interdependencies between these measures, such as cortical thickness, cortical surface area, or cortical curvature metrics. And as a result, we know a lot about these measures and how they develop in the absence of other measures. However, given the dynamic biological relationships between these distinct morphometric properties, and importantly, each carrying specific yet complementary neurobiological information, it is also crucial to study their interrelatedness in the context of typical cortical maturation. So here we propose to use a novel implementation of a multivariate technique, non-negative matrix factorization. And so basically NMF is a matrix decomposition technique that decomposes the input matrix into patterns of covariance or components and their weights. And this technique has been recently explored in the context of neuroimaging, pioneering by Dr. Sotaris and Dr. Saragenan to capture dominant patterns of covariance. And more recently, this method has been adapted and developed in our group to enable the integration of multiple structural metrics. And as in the present work, so the novelty of our work is that we've been adapting the technique to stack up the different morphometric features of subjects, and then the way which we do the decomposition gives us two matrices. So one is a vertex-wise matrix, which gives you a weight at each vertex across a number of user-defined components, and then we can look at how each of these components gets a specific contribution from cortical thickness measure, surface area measure, gyrification index measure, and mean curvature measure across all subjects that they're analyzing in our data. So based on this, the objectives of this work is to first to implement NMF, to investigate the interrelatedness of cortical morphological measures using a cross-sectional data sample, and secondly, to investigate patterns of coordinated change across multiple measures of morphology using a longitudinal data sample, and lastly, to investigate how such patterns of morphometric covariation might be influenced by variation in demographic and cognitive ability. And to do so, we've used a sample of structural T1 MR images of typically developing participants aged 5 to 25, provided from our collaborator at the NIMH. And we used 776 participants per scan for cross-sectional analyses, and a longitudinal subset of this data consisted of participants Each with 3 scans was selected for the longitudinal analysis. And notably, this dataset included participant socioeconomic and cognitive data. So moving to the NMF decomposition results, in the cross-sectional analysis, NMF identified 6 different components that we derived from our data, and each component is basically a cluster of vertices that tend to share a specific pattern of covariation across subjects and morphometric features. And here we have the subject per metric weights organized for each metric across the different components, which describes a relative weighting of each subject on each component by measure. So here you see the variation in subject weights, which are organized in an age-ascending manner per metric. And for example, you can see how cortical thickness weights are lower in older subjects, and also you can see the overall subjects variation weights across metrics. So some interference that we can make here is that, for example, component 2 is characterized by higher values of cortical thickness and mean curvature, moderate surface area variation, and lower values of gyrification index in these specific cortical regions. Whereas component 5 is more about higher surface area weights in primary heteromodal regions of the inferior temporal and parietal cortices. Moving on to the longitudinal NMF implementation, here we specifically used age-related slopes across 3 different time points derived from each subject at each vertex for each metric. And this implementation will result into a different interpretation that rather highlights the velocity of change, in which lower weights are interpreted as steeper decline of a metric, while higher weights are interpreted as a relative preservation. So looking at the longitudinal NMF results, here these components have captured vertices that share a same pattern of synchronized change across these measures, which the pattern then can be described from the weight matrix. So for example, again here, we can see that the longitudinal component 2 describes relative preservation of cortical thickness, a sharp decline of gyrification, and a decline of surface area and mean curvature in bilateral, occipital, and temporal poles and the right primary somatosensory areas. So after identifying these components of covariation across both morphometric measures and their coordinated change, so next we asked if these patterns might have significant demographic or cognitive mappings. And to assess that, so here we have the morphometric component pairs on one matrix and the demographic variables in another matrix. And what we chose to do was to decompose these further to find morphometric signatures associated with these inter-individual variations using partial least squares, which basically gives you linked patterns of brain and behavior covariance in latent variables. And here I'm showing the results of the one significant PLS latent variable derived from cross-sectional analysis that highlights maximal correlation across these 2 sets of data. And on the right-hand side, the brain maps are summarized with their specific morphometric profiles, and each significant morphometric feature contributing to this latent variable is displayed with its bootstrap ratio. And on the left-hand side, we have the significant demographic features contributing to this latent variable, which are highlighted in yellow. And so this latent variable accounts for 96% of the covariance, and it is very much an age-related latent variable, showing older ages to be associated with this distributed decreases in gyrification index, cortical thickness, and surface area throughout the cortex, and also local increases in the sulcal depth in mean curvature. And we do also observe the subtle impact of lower socioeconomic status and IQ scores on these patterns. Next, the behavioral PLS analysis of longitudinal NMF revealed 3 significant latent variables accounted for around 97% of the covariance together, from which I'm just going to show you the first. So this latent variable explained 47% of the covariance across the brain and demographic data which loads dominantly and more preferentially onto females and suggests that more socioeconomically advantaged and high IQ females accelerate through neurodevelopmental processes. And this pattern is mostly manifested in surface area and then cortical thickness almost uniquely in regions that are related to heteromodal processes. On the other hand, mean curvature changes in longitudinal component 6 exclusively shows a more stable or increasing rate of change in these individuals. And notably, comparing the results from the 2PLS analysis of cross-sectional and longitudinal NMF weights kind of emphasize that the inter-individual variability in factors such as age, sex, environment, and cognitive ability are better explained by variations in tempo of anatomical change than cross-sectional morphometric covariation. So finally, just in conclusion, we identified patterns of cortical covariation across integrated measures of cortical morphometry and their synchronized rate of maturation in typically developing youth. And we observed a non-uniform relationship between morphometric measures and their coordinated maturation throughout the cortex, which underlies fundamental neurodevelopmental processes that co-vary together in this period. And we observed that these patterns are age-related, are sexually differentiated, and have a strong association with individuals' variability in socioeconomic privilege and cognitive ability. And taken together, we believe that this novel characterization of cortical morphometric features maturation would not only contribute to better modeling the typical brain development and its underpinning mechanism, but it also shapes thinking about mechanism underlying pathological neurodevelopmental instances. And with that, I'd like to thank you very much for listening.
Thank you very much. So since she's not here, we won't take questions and we'll move to the next speaker.
So is Ming Yang in the audience or as a speaker? Because we were not sure if, so it seems not to be the case. So it's going to be on video. And the next and the last talk of the session is going to be about postnatal experience, how it shapes the thickening and myelination of the neonatal visual cortex.
Hi everyone, here is a brief introduction of my abstract named Postnatal Appearance promotes the thickening but not myelination of the neonatal ventral visual cortex. My name is Mingyang Li, a postdoctor from Zhejiang University. My present work is to answer a question that's whether any postnatal appearance influences the structure development in human visual cortex. We used the public data from Developing Human Connectome Project, the DHCP. After data filter, there are 407 subjects in this analysis. The right figure shows the distribution of the postmenstrual age, namely PMA of the neonate in our study. The distribution is nearly long, and most babies were born between 14 and 42 weeks. First, we define the anatomic mask of ventral cortex using HCP MMP atlas in adult space. The mask contains 13 full regions of interest per hemisphere and mainly include the early visual cortex, ventral occipital temporal cortex, and anterior inferior temporal area. We register this mask into neonatal space by a surface-based registration method called multimodal surface matching. The resulting mask was visually checked to assure that the anatomic location is approximately right. As shown in the left figure, the area V1 basically covered the Kerckerian circles in adult space, and you can also see that in the neonatal space. And the VOTC, the ventral occipital temporal area, covered the occipital temporal circles and the collateral circles, and it also can be observed in the neonatal space. Then we extracted the cortical thickness and the cortical myelination in the whole mask and applied the correlation analysis between the structure measurement and the PMA. In general, the mean cortical thickness and the myelination of the ventral cortex significantly improves between 13.7 to 14.5 weeks of PMA, as shown in the right figure, and the blue indicates the left hemisphere, and the pink is the right hemisphere. Then we did the correlation analysis in each of the 13 4-hours in each hemisphere to see the spatial difference of the development. As shown in the figure A and the right figure, both cortical thickness and myelination show high correlation with PMA in the posterior than anterior region, and high correlation in the medial than lateral part within the anatomic mask. This result is consistent to the previous study, but what they didn't do is to separate the effect of postnatal time in the PMA of neonates. So when we did the correlation analysis between the structure measurement and the postnatal time, we found that the cortical thickness significantly increased with the postnatal time, but the cortical myelination was not. Furthermore, we applied the partial correlation to control the PMA in the above analysis, and the effect of cortical thickness was still significant. In addition, the correlation between cortical myelination and the postnatal time was significantly negative in all RIs after controlling for the PMA. This result might reflect the impact of prenatal time on the myelination, because subjects who have a longer postnatal time would have lower prenatal time at birth with a given PMA. We further validated our results in two subsets. The first subgroup contained neonates whose postnatal time was less than 3 days, which means the subjects in this group almost have no postnatal appearance. The second subgroup contained neonates whose PMA will range from 14 to 42 weeks to control the PMA but keep the variation of postnatal time in this group. So if the postnatal time do have modulated effect on the development of cortical thickness, the correlation in the first group should be weaker or no more significant, but the correlation in the second group should still significant. And we see in the first subsample, the cortical myelination shows strong correlation with PMA, but the cortical thickness was not. And in the second subgroup, significantly positive correlation remained between cortical thickness and the postnatal time in ventral cortex, and significantly negative correlation was found between cortical myelination and the postnatal time. These results supported above findings that the development of cortical myelination depends on the endogenous mechanisms, and the longer prenatal time would be beneficial for the development of cortical myelination. In contrast, the development of cortical thickness in neonates was driven by both postnatal time and endogenous growth. Relative to term-born infants, preterm-born infants have longer postnatal time, with relatively immature development at birth, and thus the comparison between them may help us to understand the influence of early appearance and innate immaturity on cortical development. And the 14 sex RIs show significantly low cortical thickness in the term-born than preterm-born infants, and all RIs show significantly high cortical myelination in the term-born than preterm-born infants. This result shows that the early development of cortical thickness in neonates could be modulated by postnatal time even before the term-born time, which is commonly 13.7 weeks. In conclusion, our study suggests that the development of cortical myelination in neonatal period probably depends on the endogenous mechanisms, but the development of cortical thickness in neolate was driven by both postnatal time and endogenous growth. And the postnatal appearance could accelerate the thickening of neolatal visual cortex, even if it was not necessary. In the end, and thanks to the DHCP for the precious data they shared, and the guidance from the corresponding author Dan Wu, and the suggestions from the people in our lab. That's all, thank you.
So, thank you very much again to all the speakers. And again, because this talk was pre-recorded, we don't have the possibility to post live questions. So I would ask everybody who wants to ask a question to the pre-recorded talks, just contact the speakers directly. And thank you very much for the audience for attending this symposium.
Thank you.